When we raised the money, we were sort of 3 people. So we've transformed the company in the last 12 months. We'll get into the clinic next year with our first program, and we'll have the second candidate for a second program by the end of this year. It's been a busy year. I'm not sure the therapeutic index was the biggest challenge for N-myristoyltransferases. I think the bigger challenge was it's completely novel biology. When the biology is so novel, you don't know what the best cancer to go into, and you don't know how to design that stuff.
I think, biotech is just a really small world, isn't it?
I like the phrase, you need to be careful who you're rude to, because you will see them again. Never unintentionally rude.
Bienvenue to a new episode. I'm your host Philip, and on this show, I'm interviewing the best Europeans in biotech to help you grow. Antibody-drug conjugates, or ADCs, are still very hot, but new payloads are needed and are rare. One of the most promising approaches of new payloads is actually coming from Europe, more specifically from London, from Myricx. So I went to Canary Wharf to catch up with Robin. I didn't know him personally, but he was highly recommended by one of the previous guests on the show. We talked about Myricx raising one of the few recent European Series A that passed the $100 million bar to advance its NMT ADC into the clinics. We also talked about why the UK is actually pretty good at ADCs and why leaving big pharma after some time can be very energizing. So, here's my conversation with Robin, and please hit the follow button if you're enjoying it. Before we start, here's a quick word from our sponsor. Today's episode is brought to you by Cadence Science Partner, Europe's leading real estate partner for life sciences. Whether you are launching or scaling a biotech company, Cadence offers flexible lab and office space for every stages, from startup to scale up and beyond. We recorded today's episode at the Cadence London Innovation Centre in Canary Wharf, home to our guest Myricx Bio, where they've grown their company within a vibrant community of biotech innovators. With 30 plus science clusters across Europe and a global network of researchers, universities and investors, Cadence offers more than space. It's a place to grow, connect and thrive. So you can learn more at cadence.com in the link in the description below. So welcome to the show, Robin.
Thank you for the invitation. I'm pleased to be here.
Cool. So I want to start with the person who introduced us, Tim Hines from Abingworth, who was on the show before. And I've seen that you worked for him at Astex, basically almost 20 years ago, and even more. And now he/Abingworth invested last year in the Series A. So I think, biotech is just a really small world, isn't it?
Yeah, I mean, I like the phrase, you need to be careful who you're rude to. Yeah, because you will see them again. Never unintentionally rude.
That's it's cool. And so the just going to the Series A obviously was basically exactly 1 year ago, which was a huge Series A, I think $115 million, 19 million pounds, led by Abingworth and Novo. Can you just maybe go a bit into what progress you made now 1 year after?
Yeah, good progress. So when we raised the money, we were sort of 3 people. A lot of fantastic consultants, a great network of people we'd known for years. But in terms of staff, we were really small. And we were absolutely 100% focused on raising the money. Otherwise, there's no future. So we've transformed the company in the last 12 months. I think we look and smell like a very focused development company. So we've selected our first candidate, got through our first pre-IND review. We'll get into the clinic next year with our first program. So it's been a busy year.
Quite some progress there. And I've seen that the round was not easy. I guess it's never easy to close a round, especially of that size.
So Myricx, we were formed in 2019. I'd retired from GSK. I was doing bits and pieces with friends, but I was not looking to be crazy, absorbed by one thing. And a friend of mine, Roberta, said, we're gonna start this company, November 2019. Do you want to consult? Would you like to join? And I said, look, I've tracked the science. It looks really cool. I'll join. But I thought we were going to be 2 years. Then we'd raise a Series A and I'd retire. And this was for small molecules, IV or oral. And we got compelling data, but we couldn't close on a Series A. And actually Abingworth and Novo were amongst the VCs that were politest to us. They said, these are the things we really like about your small molecule program, but here's just a couple of risks. We can't get our head around both of them. So we sort of, as always, it's very good to leave on good terms. If you don't get what you want on the moment, it doesn't mean you won't get it down the road. So we parted in good terms and decided to pivot to working on ADCs. So when we went back round to raise money in late 23, early 24, our name had been around a while, and that's not always a good thing. If you're known as a company that didn't raise the Series A first time, you have to factor that in. So it was interesting. Well, clearly we were very fortunate with the timing. The world was super excited about ADCs. The world knew they wanted new payloads. And so that brought a lot of people to the table. And above all else, we're super fortunate that it looks like NMT just happens to be a fantastic payload, as long as you design and select an appropriate NMT inhibitor. So a number of things came together, but it was still tough to raise the money. This is, I'm late in my career. I'm not a serial CEO. I'm not really a CEO dude. I'm not a financial person. I'm a discovery scientist. I'm a technologist. So there's a lot of learning for me and a huge amount of help from our seed investors. So if
Sofinova was in this series.
Sofinova and Brandon, an individual of Brandon, Jonathan Tobin, was very, very helpful, both in terms of optimism and positivity, but also specific contacts. And he did a lot of work. So yeah, we got there. We got there.
Yeah, and I guess also 2023, 2024, and still today, just the investment climate is
so again, all biotech is so again very fortunate in timing. Very fortunate with timing, and I'm grateful for that. I mean, yeah, absolutely.
Yeah, cool. And I like you mentioned a bit the pivot, but going back to the story of the company, what I find pretty remarkable is that you basically 2019 the compound started as antiviral.
Yeah.
And went into small molecule for oncology and then into ADCs.
So there's a really famous drug discovery person, Paul Janssen, and a friend of mine went to his retirement lecture, and he asked him a question. He said, how did you deal with attrition in your career? And Janssen said, what attrition? Some targets take a year, some take 10 years, and some take 30 or 40 years. So it's remarkable, if you listen to a lot of friends and colleagues, they'll tell you their own stories about targets that didn't make it first, second, or third time. So NMT, N-myristoyltransferase inhibitors, was looked at as an antifungal decades ago. And one of our founders was one of the first chemists working on it, Andy Bell. So there's a lot of information that we were able to bring in and use. But the big challenge had been, if you block the enzyme, you will kill the cell. But how do you get selectivity over the diseased cell or the non-mammalian cell? And you have to factor that in. So that had been the challenge. So Myricx actually, spun out from the Crick and Imperial, and we in-licensed some fantastic IP and know-how that was based on looking for antimalarials. It was funded by Medicines for Malaria. But a key objective was you've got to get 100-fold selectivity for killing the malaria cells relative to human, and they couldn't achieve that. So they just said, look, here are all the compounds back. Here's all the knowledge. We can't see how to use it as an antimalarial. What can you do as innovative academics? Where might you take this? And that's the work that Ed Tate did to then sort of do some novel work.
And then as a standalone, then the delivery is probably a problem to the solid tumor versus with ADC, you get a bit more specificity.
Sure. So, I mean, with small molecules, there was a dogma before Myricx started that actually you just kill all cells at the same concentration. It just really would not be a useful cytotoxic mechanism. So Ed Tate screened at the Sanger before Myricx was founded, 3 structurally diverse inhibitors that Andy Bell had made. And tested it at about 700 cancer cell lines. The dogma was they'll all look the same. And the data didn't support that. The data said, we don't know why, but there's 15, 20% of cancers that are really sensitive. We don't know why, but there's 15% that are insensitive, and the rest are sort of spread in the middle. So that was innovative data. It sort of challenged the dogma of the day. And when you challenge a dogma, people say, okay, maybe these people are smart. Maybe they'll get somewhere different. So that was the data that got Brandon and Sofinova comfortable after putting a very small, very targeted seed fund.
Yeah. Okay. And then so I'm still the switch to ADCs. So you're adding target and selectivity, I guess.
Yeah, we are. So we've made and tested 700 or 800 NMT inhibitors with a range of properties and profiles. And what we found was that you could get complete tumor regression, a range of solid, hard to treat tumors. You could completely resolve them.
In animals, I guess.
In mice.
In mice, yeah.
But to do that, you needed to sort of dose episodically. You couldn't chronically dose. The animals would lose too much condition. And that's a sort of a well-known regime for cytotoxics, to dose intermittently. But one of the compounds, you needed to give sort of 4 mg per week to get pretty decent tumor growth inhibition. If you take that same compound and put it onto an antibody with a clinically precedented linker, rather than needing 4 mg per kg, you need 0.018 mg per kg. So it's hugely more efficacious. And that's what you really want to see, that actually that is a very obvious piece of data says, look, you're getting to the tumor cell, you're releasing it, and you've got far less systemically exposed. So it's a very encouraging start point. It worked. Our first experiment that we did in vivo with the ADC gave us a spectacular increase in efficacy and a very, very big increase in tolerability. So very encouraging.
Okay, both, okay, it's cool. And then, now I understand. And I'm curious, you mentioned the IP. I mean, if it was a really old compound, where's specifically the IP then?
Yeah, so as you'd expect, we've laid down multiple layers of IP.
Yeah.
So, we have patents of selection for our most preferred payloads, which are much more recent. And we've also got patents on the use of any NMT inhibitor as an ADC payload. And then we've got specific patents for specific ADCs. So we've got multiple levels.
Okay, yeah. Because what I'm just curious also on the ADCs. I mean, I've talked to a few companies. Tubulus was on the show before, and I followed Astex Therapeutics and followed a bit the space. But from my understanding, there's always either innovation on the antibody, either innovation on the linker, either innovation on the payload, either bringing the innovations to bring them together. I think for you guys, it's mostly on the payloads.
It's completely the payload. And yeah, today, mid-2025, we have really exciting preclinical efficacy.
Yeah.
We have really encouraging preclinical tox data. But it's an incredibly early project. And it's completely, novel as an ADC payload. So we do not need more novelty.
Yes.
So we try to be quite robust and take clinically validated antigens and/or antibody and clinically validated linkers.
Yes.
So we're focusing on the NMT. That is our core for sure.
Okay. And on the one thing I saw about the ADC as well is the number of payloads that you can attach.
Yeah.
And that Daiichi has basically one of a superior technology or something. Yeah.
Yeah. So it's interesting, going through the fundraising, we got to meet a lot of due diligence people from a lot of investors. And also, we pivoted into ADCs. And one of the things that we were told by, some very helpful advisors, was you don't know anything about ADCs. If you weren't successful with this mechanism as a small molecule and you're small molecule experts, how on earth are you going to cross the finishing line when you take on such a complex modality? So we just reached out to some KOLs, and when we showed them our small molecule data, they were super excited. to see what experiments they could help design, whether or not we could ask the question, does an NMT inhibitor look like a useful payload? So people were super helpful and encouraging. But as you listen to people, sometimes everybody says the same thing, but often, you get complete diversity of views. So this drug antibody ratio, some people said it's incredibly important that you really optimize that. Other people said, look, for a novel payload, I'm not sure it really matters. So we've been, I think Myricx is a pretty careful company. We're well funded. What we don't want to do is rush and take any unnecessary risks. The risk is in NMT. Is NMT a useful payload in humans? Does the efficacy translate? Does the TI translate? We don't want to step over anything and be simplistic. So we've done a pretty thorough job on DAR optimization and honed in on a specific methodology for specific DAR.
Okay.
And we'll see.
Well, and I guess you can always do a second generation and offer pass further on.
For sure. Absolutely.
I like this. I haven't heard really another example of companies even globally where they had a small molecule and then they transitioned to ADCs like this.
There are a few that have phoned me up over the last year and just wanted to have a sort of informal conversation. So there are a number, I think, of interesting biological approaches where people have bumped into TI as the issue, therapeutic index. I'm not sure the therapeutic index was the biggest challenge for N-myristoyltransferases. I think it was an obvious challenge. I think the bigger challenge was it's completely novel biology. It's one enzyme, two isoforms, one enzyme, but it blocks cells making between 150 and 200 proteins. So it's really pleiotropic. So how do you design your first smart clinical study? Which cancer, which endpoints, So I think that was probably the biggest challenge for us closing on a small molecule raise. In some ways, the industry is used to taking a risk on a TI. And they can factor that one in. They sort of know it's coming with most cancer mechanisms. But actually, when the biology is so novel, you don't know what the best cancer to go into, and you don't know how to design that study. You can't leverage a previous success. That was the big challenge. And you get that focus when you go with an antigen, because clearly that's represented on specific cancers.
That's makes sense. And can you go a bit deeper into which type of, I think it's solid tumor, which you selected, which antigen?
So we actually go public. Okay, on our lead programs will be, but they are well-known antigens. One where there is a major product on the market, this topo-1 based.
Okay, HER2 or something.
And the reason that we think that's a very interesting part about portfolio is that we have preclinical data that shows we work in models resistant to the topo-1 drug. So we can come in after you've pre-treated with the topo-1 drug and we still see really good tumor regressions. So we're building a body of data that demonstrates preclinically this mechanism, novelty of biology means that you can get responses when you don't get responses to current clinical payloads. So the biology is really the core, Myricx. We are an NMT company. We've chosen to use ADCs as a very timely way to develop our first products. But if they are useful, it will be because NMT itself is a really interesting mechanism. So it doesn't touch any of the pathways that current ADC payload use. So it doesn't touch tubulin, doesn't touch microtubulins, it doesn't touch DNA. We've done a lot of work. Our academic founders done a great deal of work to identify the key pathways. And there's a small handful of pathways that all oncologists will know, but actually nobody's actually got an approach to bring those together. So we can uniquely hit multiple targets using one enzyme.
Okay. Yeah. I was also curious that you answered a bit on how you differentiate from the whole ADC space where it's really crowded.
Yeah. So our first, one of our first two programs is a very competitive, with a really major top of one product. The other program will get to the clinic. In our first program, there are ADCs in the clinic with nothing launched, no marketed. They're the mid to getting towards late stage. And I think we're completely focused on NMT being a differentiated payload. So we're not going to try and find the rarest cancer that nobody's run a study for yet. We're not going to try and say that, ours, we win on efficacy, or, dose. We're not going to, it's not incremental. We really think the complete novelty of this mechanism should allow us to get profoundly different biology in cancer patients. In which case, our ultimate objective is to demonstrate that NMT is the best payload. So we would look forward to having the database that says in the clinic, this is a highly efficacious and safe payload and warrants coming earlier in the treatment pathway. But we all know that we're going to have to start, the clinical studies on people that have gone through those standards of care. So we are going to get patients that have been through multiple treatments, including, ADCs increasingly. So actually for us, it's going to come down to, do we work in situations where other payloads can't work?
Which is always challenging.
Though it falls slowly, as I say, I'm a scientist. I came into Myricx because I think the science is compelling. I think the team is agile. I think our investors have a great mindset. We want to ask that question: does the novel biologic really give you profoundly different efficacy in cancer patients that have seen other treatments? So that is the question that I want to ask. So I'm not intimidated by it. I think our understanding of the biology makes this an obvious and compelling experiment to run.
Yeah, makes sense. And on the curious on the NMT space as well, because I saw that there's one company,
Astex, yeah.
But they do it as a small molecule.
Yeah.
But they're in phase 2 already.
Yeah, and we know their compound well. And I think they're a very impressive organization. They've never raised sort of VC money. They've really worked extremely hard. They're very clinically led. They really believe that they can run smart studies. But how they've done it scientifically is very different to us. So, I rambled about 20 minutes ago and said that we learned from our small molecules that you get profound tumor inhibition in tough solid cancers if you treat it intermittently, 2 days on, 3 days off, different regimes. They appear to have not observed that or not learned that. So what they've done is they found the maximum tolerated dose preclinically. And then they've just translated that and found the maximum tolerated dose in humans if you dose every day for 28 days. Scientifically, to me, that makes no sense, because what we've learned is there are certain myristoylated proteins that are very sensitive to NMT. And the way that you see that sensitivity is that the cancers dependent on those myristoylated proteins die quickly. So if you only dose for a few days, the only cells that show toxicity are the ones that are sensitive to NMT. If you dose for 28 days, all cells will show similar sensitivity. So the good news is they got to the clinic and they found the dose-limiting tox to be transient GI. We've seen GI as probably the mechanistic hallmark tox that you observe, whether it's a small molecule, independent of series or an ADC. So we're learning from them, and we wish them well. I think they're incredibly focused on patients, and we wish them well. We've taken a different route. And they've I'm pondering, I don't want to say something now that I'm going to phone you up in a week and say, can you edit that? I shouldn't have said that. But you may be probably not aware, but actually they're trying to pivot to be an ADC company.
Also, okay.
They went to a meeting recently and presented on NMTs and an ADC payload. And we actually asked them, have you got data? What data are you going to use at this meeting? They said, we haven't got any of our own data. We're going to use yours.
Okay. I guess it's also good to not be, it's good to be first, but it's also not good to not be alone.
You don't want to be alone. I think I found that with investors. They realize they have to take a risk. But
want a bit of adventure.
They'd like to know the benchmarking.
Yeah, I like that. And to finish on the team you mentioned, I saw that there's 2 ex-Genmab people who joined. One is chief medical officer, one is, what is it, SV or
DPs.
APC, which I think is a very good sign.
And it was interesting. Jesper, who's joined as SVP CMC.
Yeah.
If you looked at him on paper, there's no way we were going to attract him. He's way too senior. But he was involved with he supported Novo's due diligence on us, Series A. So he got to lift up the bonnet and looked inside and just thought at the stage of his career, he wanted to do something pretty bold. He just wanted to get out of his office. He wanted to push himself. And I remember saying to him, I said, are you sure we're big enough for you? Are you sure you want to get down and dirty? Do you really want to do this work? And he said, absolutely. And
That's exciting as well.
He's fantastic fit. I mean, he just epitomizes, the culture that we need. Experienced, smart people that really want to get their hands dirty and see if we're onto something.
Yeah. And I guess it's at the end a similar profile to you as well, where you were at GSK, pretty high level at GSK.
Yeah. And I'm a technologist.
Yeah.
I mean, I had failure very early on in my medchem career. I got a couple of assets, small molecules into early development, like 5 years into my career, and I thought that's fantastic, and they both failed. And then you have to sort of sit down and say, well, when was the writing on the wall? When would a wiser, more experienced person have done something different? And, so I did a lot of reflection. And actually, in hindsight, they were high-risk compounds. And so I became very interested in causes of attrition, molecular design, physical properties. So most of my career has been focused on maximizing the chance of a specific project working. Trying to be ruthless about when you've got too many risks. And that's probably why I did a number of the jobs and why I went to Astex. Astex had no money, no labs, no chemistry, but a compelling idea. And that just appealed to me.
Yeah, I like that. We'll transition to, or maybe your last question on Myricx also is that I saw that Chris Martin, also the founder, CEO of Astex Therapeutics, and probably really big guy in ADC, at least in Europe. He's a chairman, sure.
And
it must help, I guess.
For sure, on many, many levels. Above all else, he's a genuine person that wants to contribute and help the team. So, he makes himself very accessible. He really wants to help. But clearly, this was pre-Series A raise. I was being pretty ruthless. I needed somebody to increase our credibility that we had something worth investing in. It took me nearly a year to get Chris to join. And that was the highlight of my 2023, was getting him to sign as the chairman at the end of the year. We didn't have the Series A at that point. But when I spoke to him at the beginning of the year, I said, this is what we're looking to achieve. And this is what we're looking to achieve this year. And he just said, well, ring me up when you got those milestones, keep speaking to me. And so it took me most of the year. And by the end of this year, he said, okay, the team seem to do what they say they're going to do. The data seems really, really encouraging. I'll join you. And I think he's come for fantastic reasons. That he's learned a lot about ADC companies. He's learned a lot about being a senior exec. And he's learned a lot about how to sort of keep your feet grounded, to really focus on the key things when you're developing novel payloads. The whole world wants novel payloads, but most of the world knows they're incredibly hard to discover and clinically validate. So I think our board, investors, and our chair, really walk that talk. We're optimistic, but we're also ruthlessly objective. We've got to do critical science and get that data.
I can see also from, let's say, some kind of a takeaway, but also from everything you mentioned, that you are trying to really isolate the risk, not take too much risk, not do something too crazy fancy, but just the biology alone is already, I guess, very risky. So isolate.
Yeah, absolutely. As an old chemist, you need novelty, but you don't want too much.
Yeah, I like that. And how, curious also if you had another lesson. I guess you learned a lot of lessons across your career, but maybe recently from whatever your last 5 years at GSK, is there one like lesson, especially in drug development, that struck you?
Yeah, I mean, so I was at Astex for 6 or 7 years, small biotech in Cambridge, UK, and it's fantastic. Probably
Successful, like
the best 5 6 years I'd ever had. But I wasn't as confident as I wanted to be that they would find drugs. I was unbelievably confident they had brilliant technology and they were creating really good leads, but I wasn't sure they'd be a standalone drug discovery company, and that's what I wanted to do. So I surprisingly, I moved back into big pharma, into GSK, and most people don't go back into big pharma. And I did, and I'm glad I did. We had a brilliant few years at GSK when it was very focused on respiratory. And then your company reorganized a number of times, as you need to.
Yeah.
And I chose to take a senior role in platforms. And I guess I hoped that I would be experienced enough and smart enough and good enough to be helpful, across dozens of programs. And I think what I realized was I couldn't hit the level of scientific impact that I wanted. I couldn't be close enough to the data. I couldn't in real time nudge people and help them solve problems. So I ended up as a sort of strategy manager, portfolio budgets, and I got no energy from it. So I was there for 11 years. I had 7 jobs. And when they asked me to do job number 8, I just had to be honest to myself and the organization that I needed to go somewhere that was going to give me energy. So I guess my honest. Oh, sorry.
That's honest. Yeah.
I am honest.
That's good.
I'm certainly not always right. I try to be honest. So it was with a heavy heart that I left GSK. I'd have loved to have been able to be more useful to the organization. I did my absolute best, but it just wasn't energizing for me.
Yeah. I've seen also, it makes me think also, I mean, on the show, there's a few people who came from big pharma and moved more to the biotech space. And I think it's a theme that came a few times, like either they were, as you just said, maybe less energized in big pharma and more in biotech, or just they had a more exciting opportunity in biotech.
They're different organizations. I mean, they just suit different people. I'm not putting a value judgment on it. They are different. And I think
It's different phases of career.
I learned most of my science in big pharma. I've learned most of my experiences, and I'm extremely grateful. But at this stage of my career, I want to go around it one more time to do something really novel, really innovative, that's high risk, but if it works, incredibly high return for patients, investors, for everybody. So that's just what I want to do, and I don't want to spend too much time on breadth of things.
Yeah, makes total sense. And I'm curious on the, like, from what you say, you sound more. I mean, it sounds like you're energized by the whole mission and everything. Sounds like it fits you well, although
it does fit me well. I mean, it does fit me well. It's a compelling experiment to run, and I want this team to run the best possible experiment, to be very capital efficient, to be really focused on what success looks like. Success isn't just dosing it in the clinic. It's got to be impactful. You've got to really position this payload. You've got to really anticipate what people will require. So it's a compelling experiment, and I feel super, super lucky at this stage of my career to sort of bumped into the opportunity.
Yeah. And how was it for you, or maybe if you can look back to other people who want to do something similar to you, maybe a senior role, and then they want to run a company or a small biotech. How was it for you, like to transition? I think you didn't start as CEO, but then you became CEO, right?
Yeah, I don't aspire to be the CEO. I aspire to be part of the team that does great things. My friend Roberto, one of the 3 founders, was the founding CEO. He's a small molecule person, but he's a biology person, and he just felt ADCs were very chemistry, different modality. He said he was ready to just step back, become a consultant. So, I stepped into the CEO role because I wanted this to succeed.
Yeah.
And so I just cracked on with it. But I think now that we've raised this Series A, now that we've got a couple of programs with really clear paths to the clinic, I believe now's a great time to bring in the next type of CEO, somebody that will really grow the company, that's really far more development, far more commercial, and far more US and world known. So, I'm working very hard in this role on many levels. One of them is to add to the team somebody else that's outstanding.
You're looking for someone
that will, yeah, absolutely. And I think that's super exciting.
Okay. And then you would stay in more on the scientific and discovery road, I guess.
I think you picked up on that. My passion, is that. And we've got a couple of great programs. We're using the same linker payload. So if both of them work, that'll be incredibly bold and incredibly successful. But I want to lay down options should we want slightly different profiles with the linkers, with the payloads. So I want to do some skunk work. I want to do some stuff almost behind the scenes that won't touch our first couple of programs, but it's going to give us breadth, it give us resilience. And to be frank, when people look at the organization down the road, they'll say Myricx are really strong in multiple ways. Their clinical programs, their platform, their breadth, their level of understanding. So I want us to be excellent.
Yeah, I like that. And I like also, sounds like from everything we discussed, because you have so novel target and biology that you can really build this platform and have this breadth.
So we've, as you'd expect, we've done a comparison of our payload to DXD, the leading ADC payload.
Yeah.
And we've looked at sort of, 100 odd cancer cell lines. We have a very similar breadth. We're at least as broad as Topo1. We have very comparable IC50s for major cancer types. So, the breadth of opportunity is immense. If the novel biology can start to indicate in the clinic that we work where Topo1 doesn't, and that we have a very clinically manageable therapeutic index, the scale of opportunity is truly immense. And one of the things that we've learned is that, yes, we have a novel mechanism and great efficacy. We've also got novel tox profile. So we do not show that so far we have not seen the tox that typically limits the use of current payloads. So we don't see ocular tox, we don't see neurotox, we don't see lung tox, we don't see impacts on neutrophils, we don't see neutropenia. So actually these are the things that often compromise the dose that a patient can take, the duration that a patient can take it for. So that novelty of the tox profile for me is as exciting as the novelty of the efficacy, because it's often the tox that impacts and limits how thoroughly patients are treated. So at a very high level, I think ADCs are truly remarkable in modality in terms of response rates. Very, very high response rates. But the duration of response is not as strong. The large number of patients need dose holidays, they need dose reductions. And it's because you're just walking that line between
efficacy and tox profile.
So, I really hope that this novel mechanism will have a novel profile in the clinic, and that we will be able to fully engage the mechanism and have a much lower the tox to the point they're not compromising dosing. So I'd love it if this payload actually can be dosed to fully engage the mechanism throughout the duration of the patient treatment. Wouldn't that be a great experiment to be of? And we'll see. But if you don't, I think you don't have clear, articulated, ambitious goals.
Yeah, and I guess you will see in your phase 1 and testing in humans. So we'll see. It's a big step and a different step than translating from animals to human is always a big challenge. So maybe staying on the personal lesson, leadership lesson. One thing also is always, I think, a challenge is this transition from academic data to being a biotech.
Yeah. So we were super lucky. So Myricx have had luck in a number of ways, timing of funding, into ADCs. The other thing was one of the 3 founders of Myricx was Roberto Solari. He was also a co-founder of Astex.
Okay.
And I've known him for decades. And actually, I was having such fun at GSK when I was head of chemistry and respiratory that, I brought him in to work with me at GSK. So we worked as colleagues at GSK. He he then decided to go to Imperial, late in his career, because he just had a passion to push some respiratory virus work. So actually, although it spun out of Imperial, it had Ed Tate, world-class academic, knows a huge amount about NMT, world leader. It had Andy Bell, an ex-Pfizer chemist that chose to go to Imperial rather than go to the US with Pfizer. And then it had Roberto at Imperial. So actually 2 out of 3 were proven, drug discovery people. And also Roberto, a serial entrepreneur. So it was just a fortunate collision.
Okay, yeah. Which is quite rare.
So, I mean, I've been the other way around, where I've signed quite significant collaborations with major academic names, and we haven't been able to translate their beautiful academic assays into something that can support drug discovery programs. So I think we've been super fortunate.
Yeah, for sure. And besides experience and besides the people, like, are there any, like, let's say, some practical lessons on the translation? Like,
we've come to the ADCs sort of, in its second decade. So there is a lot of experience out there in the world. So I think one of my lessons has been, if you have one thing that's interesting, be prepared to share that with people. Because if you've got something interesting and they can add to that, people like to add to it. So when we showed them our small molecule NMT data, bioconjugation scientists want to contribute. People that have retired from major ADC companies want to contribute. So what I've learned is, if you've got something exciting, even if it's a small thing, other people want to join and see if they can, as a team, do something useful. So it feels like a team, and that for me is really energizing.
I like that. Yeah, especially I've seen that quite a few times. Especially I think with younger biotech entrepreneurs who come from academia, were like, "Oh, my idea is so precious. I should not share it. I should do my own thing," which I think is something. I guess some people will lose quite some time and don't get the best results. So it's basically.
And I do some mentoring for academics looking to do translation, and you see the whole repertoire.
Yeah.
Some are very open and some are very defensive. It's just how people are, right?
Yeah. And going a bit more into the industry of the ADC industry, one thing you said, and what's pretty interesting as well, was that you said that the UK / let's say Europe at large was pretty strong in ADCs. For example, Astex Therapeutics, AstraZeneca has a huge amount of experience.
Arvaris, Biblis, Mablinks. I think, there's significant successes of innovative ADC work in Europe. As always, the US has a huge scale, but actually China.
And Japan, I guess, also.
Sure. Yes, some very, very good individual companies. But, just as I think China's had a long-term strategy for EVs, electric vehicles, it almost felt it couldn't compete with petrol engines and diesel engines. So it made a big long-term strategic play. I think the same's true with ADCs.
And biotech in general, yeah.
Well, I think they clearly committed a long time ago to high-end innovation, structural biology, etc. But ADC is remarkable. So where do I think the competition will come from? In the last 6 months, we've heard on the grapevine of 2 startup companies very early in their work, but both saying they want to work on NMT as a payload.
From China.
One of them is an extremely high-end US academic, serial entrepreneur who always attracts money and really, really smart people. And the other one is a very good Chinese ADC company. So I think, at the moment, I think we've got an advantage, maybe 2 years, maybe 18 months. But people are working on this, so I think, how do we respond to this? I think for us, it's to stay focused and to at least meet our timelines to the clinic. And if we can exceed them, let's exceed them. So I think we've got to keep moving forwards. We're building the team very successfully. We've got to keep incredibly focused on timelines. We've got to be operationally excellent. So I joked at the beginning that, before the Series A, we're all about raising the money. Now we've really become a small but focused, company that must develop these into the clinics. So I don't want to be beaten. I mean, I don't want to, we don't want to be known as the innovators. We want to be known as the people that translated this successfully to the clinic. And if we ask the question and NMT just doesn't work in the clinic, I'll be gutted. But as long as we've asked a definitive question, then scientifically, you just got to accept that.
And I like that. Yeah. And the what's the order of magnitude when you say like US at the scale or China in terms of ADCs? Like, if you compare.
That's a very good question. Depends how you cut it, because I think quite a lot of the Chinese work is in major CROs, and some are more independent companies. I think there are far more scientists doing ADC work in China than there are in the US.
Wow.
Far more. Far more. But as always, innovation is critical, and Europe, the US have great track records.
Mm-hmm. That's good. And what's your take on the ADC market in general? I mean, I think it's grown like crazy in terms of. I mean, I think I saw a figure was it 2023 was above $10 billion revenues, and it's expected to grow a lot.
Yeah,
like
I think 15% annual growth.
Yeah.
I mean, clearly I read the reports, I look at them, there are hundreds of ADCs at different stages of development. Do I know them all in detail? No.
Yeah.
How do I contextualize them all? For me, the context is, at the moment, many ADCs give a good response, give you a good response rate, but actually duration response is always a key vulnerability. So, for us, I just think we have the potential to be very clearly differentiated. So if NMT translates, we will have clear differentiation against all of those ADCs, is my personal view. So I think it's a good time to have innovation and to be differentiated. I mean, but there's so many levels of smart work that people are doing in ADCs. We hope we're doing very high-end payload. There's other people doing fantastic work on linkers, fantastic work on release mechanisms and designing antibodies. And then there's bispecific antibodies, dual payloads, even 3 payloads. So I'm humble, and that we're just one group trying to do smart work, but we are really focused on unique biology and seeing if that translates. If it does, then I think we'll have huge opportunities.
You cannot do everything anyway.
And we can't, one of the things I learned, when I was at Astex, 3 of the programs it worked on got partnered really early. And they're the one, they translated into 3 launch products. So I'm very fortunate that I've seen.
And how many were? I think 2 were blockbusters.
I think a couple of, So I mean, one of them is, multi-billion, CDK4/6. So, I mean, my lesson from that is that Astex did something unique. They did the fragment screening. They found high quality early leads. They had a toolkit that could iterate and optimize their leads, but actually it partnered early because it knew that it needed other things. It needed resilience, tenacity, but also needed development expertise. And actually 2 of those 3 products were developed by first intent as combinations. So they were launched using an asset that the major pharma already had. So as we progress, as we go through the risk discharge, as we go through the value increase, the whole range of opportunities for us to really amplify that success by very smart strategic partnerships with one or a small number of strategic partners. So we won't be able to do it all, but I think what we've got to do is we've got to ask the question, is NMT a really valuable payload?
Yeah.
And what we've learned is not all NMT inhibitors are the same. You can't take any NMT inhibitor and get efficacy on an ADC. You can take payloads.
The devil is in the details, I guess. Yeah.
It is. So we've taken some of our own that are sort of like a single-digit nanomolar. A medicinal chemist would look at that and say, that's really potent, that's a great compound. You stick it on the ADC, you don't see efficacy. So the rules of the road.
Yeah. And I'm curious on a bit on you say, but I think investors, as we talked, there's a big appetite, or there was a big appetite in ADC. I don't know now, maybe it's a bit more crowded. I guess from big pharma as well, you had a very strong appetite when they see the sales number and they all want to go in. How do you still see it like today? How like
I think
do you see the appetite?
strong interest in investing in differentiated ADC opportunities, but clearly the bar is very high.
Yeah.
I think in terms of pharma, I think their appetite remains. Because we've raised very good money.
Yeah.
Because we've recruited some very proven, talented people around development. I think they realize that's what we're focused on doing. We're going to translate 2 programs to the clinic. So I think, we're finding a lot of pharma want to get and stay close to us, and just, and see how far we get.
Yeah.
Can we keep chipping away at risk discharge? We're having those conversations, most of them not under CDA. 1 or 2 where we think the companies have a unique insight, or we really would value their judgment, we're going under CDA. So my objective for this year in terms of BD is engagement, very mindful engagement, to bring people along, because it's very important they get familiar with NMT, they get familiar with Myricx. We don't want to just email them something and say, look, this is what we've got in that first X patients.
Do you want to partner in 1 month?
Yeah, we want to bring them on the journey, and that takes some amount of time, but it's also energizing.
Yeah, I like that. And I guess I heard that a few times on the show. At the end of the day in biotech, you want to have a lot of optionality, and you want to have whatever fundraising, capital raise. You want to have partnership optionality, and you want to push everything as much as you can.
So
I guess that's part of your job as CEO also too.
It is, and I think part of my job as a current CEO is to bring in the next CEO that actually has an outstanding track record of going to the next level. So we will be very active in that regard. In terms of long-term planning for funding, we can't assume that we're going to knock the ball out of the court, with our first phase 1A dose escalation. We don't want to, we don't want everything to be hinging on like 1 patient.
Yeah.
So
I like them. Yep. I think that's a great wrap up to thing. I want to just finish with some like a quick fire, just some quick questions, a few minutes, and then just some quick questions. 1 sentence answer. First one, what's on top of your mind at the moment?
Sino GLP readout for our first program. I just want that data. So the dose range finding, the report is 700 pages. I want to get the data and want to get that report. That's going to be absolutely transformational.
Yeah. 1 advice to young life science professional who want to, grow.
I'm not sure I will. I'm not sure I could. I think I'd just say, try and get themselves into a position where they get as much energy as they give. But that's easy for an old guy to say, right? When you've got kids and you've got a mortgage, there's Breslow's hierarchy of needs. So sometimes you just got to focus on squirreling away the money. You got to I'd respect anybody who's just trying to find their way through. So just don't look to too many people and just crack on.
Yeah. But I like the finding the fit of what energizes you. I think it's
what they say, if you find something you like, you never work a day in your life.
Yeah. What's one of your favorite biotech or science book?
I'm dyslexic. And so actually I don't read books for pleasure.
Yeah. Okay.
I'm very visual.
Yeah. 1 mistake you made in the past 12 months.
Last 12 months, crimes. I'm struggling to think of a big mistake in the last 12 months, honestly. I can give you some big ones. I think we've had a good run of luck. I can't think of one.
Well, it's okay. 1 new habit that you adopted recently.
Embracing new media.
Like a podcast.
Like a podcast.
And last one.
Actually, ChatGPT, AI.
Yeah.
Phenomenal fun. It's like having 6 buddies that are okay if you ask them terrible questions.
Yeah.
So you can just triage crazy questions. I love it.
I have to say on this, I mean, we could talk a lot about that. I love also for the biotech audience, how good it is when you ask super specific questions. I'm surprised. Sometimes it's like, I mean, you have to know what comes and what's true, but like sometimes it's.
I think if you're asking specific questions, you've got to be prepared to then do the legwork to track down the references, because there are a significant number of inconsistencies. But if you're just trying to get your brain working, if you're just trying to contextualize things, it gives you that terrain almost for free. And then you can say, that's not worth thinking about, or that is. So it's just like, I feel like I have dozens of ongoing conversations with ChatGPT. I run the same type of search. So that I love it.
Yeah, that's great. A last question. Who is one of your biotech pharma heroes?
Biotech pharma.
Or mentor or hero? Yeah.
Yeah. So I've been super lucky to work with and report into some amazing people. But one that sort of, I only realized how invaluable he was many years later was a guy called Alan Baxter. He was a very senior person. He chaired project reviews at Glaxo Wellcome. And when my first program, when our 2 assets failed to reach the clinic, I asked the project team, have we tried our best ideas? Should we keep this going? What do you recommend? And we all said we've done our best ideas. We haven't got other good ideas. So I proposed to the management council to close the program. And it was in the days you walked in, you gave your presentation, then you walked out. It was very formal. And as I walked out, somebody tapped me on the shoulder, and I looked around, it was Alan Baxter. And he said, I just want to shake your hand. I said, why? He said, you're the first project leader that's ever come to my review committee and recommended closing their program ahead of me having a coffee with them. And I didn't realize what that meant, but what he was saying was actually he and lots of people really value people making those tough calls, making early calls. Don't hold on to things. Be confident that the next thing will be even better. Be confident what you've learned will stead you well in the next position. So I've never been frightened of closing projects. I've never been nervous about going to ambiguity. And it's because of people like Alan. He's not the only one, but it was a small thing. But that permission to fail is very, very empowering.
Yeah, I like that. I think it's a great way to end the conversation. Thanks a lot, Robin.
Okay.
Yeah.
Was that okay for you?
Very good. I'm impressed by the story behind Myricx, especially the persistence to reuse MNT and the opportunity to combine it with ADCs. I'm also impressed by Robin's clarity of his role and his limits. If you've also enjoyed this episode, please hit the like, follow, or review button. Any of this action will help us a lot. If you want to support the show, you can make a donation by clicking on the link in the description. If you want to see similar videos, please feel free to check out our channel where you can find many more. I would also be curious to hear what you think. So if you could leave a comment wherever you are, or shoot me an email at philip@flot.bio. All right, thanks for staying until the end, and catch you in the next episode.